And welcome back to Pre-Health Spotlight's interview series. Today we will be speaking with Dr. Melissa Kane, an assistant professor in the Department of Pediatrics at the University of Pittsburgh. Thank you for joining us today, Dr. Kane.
You're welcome. I'm happy to be here.
All right, so to get the ball rolling, could you tell us a little more about your position here at the university and the type of work that your research group does?
Yeah, absolutely. So I joined the faculty here in January of 2019, so coming up on our 3-year anniversary of the lab. And we're interested in retroviruses and specifically how hosts defend against retroviral infection. And we're interested in lots of different retroviruses. So the most famous one is HIV-1, and we study how cells directly try to defend against HIV infection. And that's all in vitro molecular virology, molecular biology, cell biology work. But I'm also interested in how immune responses like antibody responses and T-cell responses are generated against retroviral infections. And to investigate that, we use mouse models of retroviral infection. So these are actually distant cousins of HIV that naturally infect mice. And the two viruses we use for that are mouse mammary tumor virus and murine leukemia virus. And they cause the diseases that it sounds like.
That all sounds very fascinating. Is there a specific project, either ongoing or planned for the future, that you're especially passionate about or excited about that you'd like to share?
Yes, there's more than one, but so one of the things that we're interested in is this specific antiviral protein called MX2. And I identified this protein along with 2 other people at about the same time when I was a postdoc. And this protein is expressed when cells are stimulated with interferons, which are basically like the alarm system that a viral infection is happening. And what happens is it sits at the nuclear pore complex and blocks the virus from passing across the nuclear pore to get to the cellular DNA. And we're trying to understand how it interacts with the nuclear pore complex to block HIV infection. And we're using CRISPR-Cas9 technology to make really precise manipulations in the nuclear pore complex in human cells. And the reason that we're really excited about this is that it's allowing us to make really fine changes to this really complex structure to really pinpoint exactly what interactions are happening. And this is only something that we can do with CRISPR. This is a newly available tool for us. And so it's been really exciting to apply this to fundamental questions in HIV biology. So that's a project that we're really excited about right now.
Awesome. Thank you for sharing that. So working our way backwards a little, could you tell us about the steps you took starting from the beginning of your undergrad education to get to where you are now and how you discovered your passion for research?
Yes, absolutely. I actually really like talking about this. So when I was an undergrad, I was actually an animal science major. So I know all sorts of nitty-gritty details about animals of economic relevance. And I was an animal science major because I thought I wanted to be a veterinarian. And that goal kept me focused on my education and got me taking a lot of science classes. And however, I never really thought that much about what it meant as a job until I graduated from undergrad and was admitted to a very good veterinary school and freaked out and realized I didn't want to go and didn't know what to do with myself. And a professor that I had gotten along well with suggested to me that he thought I might enjoy research and that I should apply for research technician positions. And it took me some time to get one because I didn't do research as an undergrad. I had my laboratory courses, but I didn't work in a lab outside of that. And so a lot of people were hesitant to hire someone without any experience. So my first job was actually doing research for product development for a fiberglass company. So we would go to this plant in Georgia and make changes to the way that the fiberglass was being generated and then run tests on it, which was interesting, but I had bits of glass in my skin every day, and it was certainly not what I wanted to do long term. But then I got a job at a immunology lab at the University of Colorado, Denver, / National Jewish Medical Research Center, working for a woman named Roberta Palanza. And there I studied B cell development, which is, as a basic science research, understanding how cells go from being a stem cell to being a B cell that makes antibodies. And I very quickly fell in love with research and knew that it was what I wanted to do forever. And Roberta was incredibly supportive of that as well. She told me actually very quickly after I joined the lab that I should apply to graduate school. And she was actually saying that I should do it that year, and I didn't feel ready. And I waited another year. And then I really didn't know exactly what I wanted to study for graduate school. I knew that I thought the immune system was interesting, and I knew that I thought that viruses were interesting, but I didn't have a concrete idea of what I wanted to do. So I applied to a large number of PhD programs and interviewed around, and ended up at the University of Chicago, where I joined the laboratory of Tanya Golovkina, studying genetic and immunological mechanisms of retrovirus resistance in mice. And I really just loved working in the lab, loved being a scientist. She's an amazing mentor. And we're actually still in touch. My group has lab meetings with her group over Zoom, which has been fun. And then I decided that I wanted to learn molecular biology, and I joined the lab of Dr. Paul Binosh at the Aaron Diamond AIDS Research Center / Rockefeller University, to study intrinsic defenses against HIV and other retroviral infections. And I was actually in Paul's lab for about 7 years before I accepted the position here at Pitt to run my own lab. So that's my road to the Kane lab. And I have to say that that's a really weird thing to say still. And the glassware has my name on it, which is wild. I assume one day I'll get used to that, but
yeah,
feels new.
let's talk about that. How has the transition been from doing research in other people's labs to, being the boss, the head honcho, calling the shots, really directing your own projects and kind of doing whatever you feel like you want to do?
It's pretty wild. So, I mean, one thing I love academic science, and it was actually challenging for me to get an independent position for a number of reasons that are not all that interesting, but you are not really trained in the how to run a lab aspects of it. You're trained as a scientist. And then all of a sudden, there's a pile of money that you need to spend wisely, and you need to hire people and train them and manage them. And it's a bit of a roller coaster. And I mean, for me, one of the things that, there's just so many things that are, sorry, I'm stumbling here a little bit, that you learn to manage. So one thing is, for example, you have to be able to divide your day into smaller chunks. So ideally, when you're a postdoc and you're working in someone else's lab, if you're writing a paper, you can devote a day, a week to writing, and that's your writing time and your dedicated time. And then you have time where you're dedicated to experiments. But now I have to juggle that and meetings and helping people in my lab troubleshoot and reviewing manuscripts for other people and doing things like this podcast. And so you have to learn how to switch your mindset very quickly so that you can utilize smaller chunks of time for things that you used to have larger chunks of time for. The other thing is obviously just learning to manage people and learning to really accept and appreciate individual differences, and that everyone is not you and everyone doesn't think like you. And to step back and say that it's okay that I'm the one who cares the most about this lab, and it's okay that my research technician isn't lying awake at night thinking about this project. That's not their job, that's my job.
Would you say that your past mentors will have an impact on how you yourself will mentor students going forward?
1000%, yes. And my past mentors, Tanya and Paul, are very, very different. They're both very serious and very intense people, but Tanya is very heavily involved as a mentor and very active and even pushy about it. But I mean that in in a good way. And with Paul, you really have to come to him if you want his help. But he gives back everything that you put into people. And also the expectations that they both have are very high, but how to learn to manage my own expectations for people versus managing their capabilities, that's also informed by my mentors. I don't think it's possible to have a completely independent mentorship style that's not influenced by the way that you were mentored.
Gotcha. Going back to what you said a little earlier about your path, most of our audience are undergraduate students, and I have to say personally, as someone who's on the verge of graduating, I derive some comfort from your story of like you had a certain plan, but you completely shifted gears and you worked for a little bit and you discovered your passion while you were in the trenches, not necessarily something that you like had in mind all along.
Oh, absolutely. And I would actually, to your audience, I feel like there's so much pressure to know what you want to do right when you graduate college. And I think that's kind of crazy because it's also really different being a person in the world than being a student. And I found it was really, really valuable to have time where I had a job and I wasn't a student. I didn't have homework. I didn't have exams. I had a different set of responsibilities. And it gave me the time to think about what it is I actually care about and what it is I actually want. And then I went back to school. And graduate school is very different than undergrad, but it still is school. And I think it was, not everybody needs that break. I think a lot of people do. And I've actually would also say that it's hard to let go of an idea of what you thought you wanted to be. So I was so sure I wanted to be a veterinarian that I never thought about what that meant or whether or not I actually wanted to do that from when I was 8 to when I was 23 and actually facing the decision. And I've actually had 2 technicians in my laboratory start very sure that they wanted to be medical doctors and are now applying to science graduate school, which I personally feel very proud of, but also just an example of, maybe when you try something different, you'll realize that your interests aren't what you thought they were. And it's so common for people who are interested in science to think, oh, medicine, which we need doctors, like go to medical school if that's your passion, but it's not for everyone.
So building off of that, if you had any piece of advice for undergraduates who are set on a career in academia, what would that advice be? Like what kind of opportunities they should seek out, what their mindset should be, things like that?
Yeah, I mean, this is a tricky one because I feel that there's a lot of emphasis on work-life balance lately in the culture, especially on like science Twitter, which I think is great and important. But to be perfectly honest, you have to give a lot of yourself for an academic career in science. It is a commitment, and there are challenges to it that I think are also a little bit unfair. Before I get to, I think, the good parts, I love it, I will say. For example, geography can be a problem. So if your spouse or partner is also a scientist, you're going to have to find positions at the same geographical location can be a challenge. So just being aware that it's a commitment that's it's different than a lot of other types of jobs. But get experience working in research labs, number one. As an undergrad, after undergrad, apply for technician positions. Tons of people are hiring technicians. Try to find something that maybe the description of the research excites you. And then just keep thinking about what it is about science or what area of science excites you. And then just keep seeking out opportunities to pursue that, because people in my job really love training young scientists. This is why we're academic scientists and not working at companies. And so if you're seeking out these opportunities, you're going to find people who are going to foster that interest in you. And then the other piece of advice that I would give is that while you're doing your studies, while you're doing your PhD or your technician job or your postdoc and preparing, if you really want to have an independent research lab, I think it's important not to look at those as things that you have to overcome to get to another goal. It's easy to think about your life as being something that you're waiting to start rather than remembering that what you're doing right now is your life. And so it is, just to go back, that even though it is a demanding career and you're going to put in a lot of time and hours and sweat and tears, it is important to have that balance and enjoy it as much as you can, because this is all part of the process. And I found actually my PhD years and my postdoc years were overall really wonderful time, even the difficult parts. So, and I think part of that is because even though I wanted to have an independent position, I didn't look at them exclusively as steps along the way to get that.
That sounds like great advice. So again, for those people who are considering this path, from my own personal reading, I found that there's a lot of people who are graduating with PhDs in biomedical sciences, but not necessarily the same numbers of openings in terms of postdoc positions and faculty positions. So for people who are kind of staring at that mountain from the ground up, what would your advice be for trying to navigate that uncertainty and, stay on track?
This is a big problem. So I guess, first of all, I think academia has really failed young people in this regard historically, and we're just now starting to address how to deal with this problem. So one aspect of it is that academic scientists always want to train academic scientists, and then have sort of pretended like there were as many faculty spots as PhDs that we were training historically. And that's just not true. It's just not true. You have to have a certain type of level of success and a certain type of personality. I don't even know if that's the right word, but not everybody is going to have an academic position. And I think it's really important not to look at not having an academic position as a failure. I don't even like the description of non-academic careers as alternative. There are a lot of things that you could do with a PhD, and they're not lesser than an academic career. So, looking at that mountain now, I guess the way that I would try and look at it is I would really like to have an independent career, but there are other reasons that I want to do this PhD. I really want to do an independent research project and develop my critical thinking skills. And then if I do pursue an academic career, great. If I pursue another career that utilizes that education, also great. And so one thing I think that a lot of programs that are doing now, including the program in microbiology and immunology here at Pitt, is really making some effort to foster interest in other careers that PhD scientists can do. And we're actually starting a new program here that's gotten some funding from the Burroughs Wellcome Foundation for exposing students to other career paths and connecting them with people that are actually doing those jobs. Other jobs that people don't necessarily think about are editor, a scientific editor. I have a friend who's a scientific editor, and that requires a PhD level of experience. Some appeals for him are that it's a 9-to-5 job and he doesn't have to take it home with him in the way that I take my work home with me. I have another friend who works in intellectual property law. So he's actually an in-house counsel at a law firm in New York that specializes in patent disputes, which I would think maybe would be a little bit boring, but he loves it. And he spends his time reading and thinking and investigating whether or not something someone has tried to patent is actually novel. And he said he utilizes more of his education in that job than he did in his postdoc. There's also science policy. There's also teaching at an undergraduate level or like a really fancy private school for at the high school level. There are just so many things that people can do that are not an academic career. And I think it's actually on us as faculty and as mentors and training these young people to make sure that we don't expect that we're only going to have people who want to have an academic career in science, but to do whatever we can to help the people that do.
Again, great advice. Thanks for sharing. So to round out today, I wanted to circle back to an article that some of our watchers today might read on our website about animal ethics in research. So I'm sure that your interest in this topic was started in undergrad with your background in animal science, and then one of our writers, Carlos, wrote an article on animal ethics and research. Did you want to share anything about what you spoke with Carlos about when he was interviewing you?
Sure, absolutely. So Carlos had a lot of questions for me about, he actually started in an interesting way asking me about what is life, which I remember being an undergrad and finding an interesting question. And unfortunately, now that I'm an old person, I don't. When we talked a bit about, how these things are regulated and what not only we're expected to think about when we embark on animal research, but also what I personally think about when I embark on animal research. So, because it's not an easy question, we're sacrificing living creatures for scientific questions. And for me, the baseline is really that I better believe that this is worth asking this question, that this experiment is worth doing, that this sacrifice is worth it, or not in vain. And then also a lot about the way that we are continuing to evolve, the way that we think about how these animals are housed, fed, cared for. These are constantly evolving and important questions. And there are a lot of things that we used to do that we don't do anymore because we now understand a lot more about animal behavior and animal perception. And so those are some of the things we talked about. The other thing that we talked about that I think it's important to emphasize is that these things don't happen willy-nilly. The questions that my lab asks using mice, it's because we can only ask them in mice. There's no simpler organism that we could address them. And I think that they're important questions. And we are always asked to prove that the question that we are asking in the lowest quote unquote organism that we can go to. So I could be doing my research in a chimpanzee, but do I need to? No. And would I want to? No. I don't know if there are any other specific questions you wanted to ask in that area, but.
Yeah, I just wanted to give our listeners an overview of what that article might consist of. And there's a lot more detailed discussion of it in that artiple if they wanted to.
Yeah, I really enjoyed talking to Carlos.
Yeah, speaking of discussions I've had with Carlos, I actually wanted to end on a fun note. He was talking about how much the bacteria that he sometimes grows in the lab might smell, and how there's discussions of whether the lab in your bay or the bay next door smells more.
We smell them. We smell his lab because of the smelly bacteria.
Gotcha. So
my lab is not smelly. We don't have nasty smells. Well, except for when we have to bring the mice up. So the mice are a little smelly. But
right. So I guess the moral of the story is if you can't handle smell, don't work with bacteria. That's what I'm getting from Carlos.
Definitely stay away from bacteria if you don't like smells. I'm actually really sensitive to it. And when I found out that my nearest neighbor lab was a bacteriology lab, I was a little bit bummed. But they're great people, so I'll let them have a pass.
It balances out.
Yeah.
Good. Awesome. Well, that's all the questions I have today. So thank you so much for speaking with us today.
Great. It was great talking with you.